Welcome to my talk. It's my great pleasure to get the chance to talk about a matter of the heart and present my work on simulating and researching heart diseases and treatment options in 3D heart models. I want to highlight once again how thankful I am to the Jung-Stiftung for being awarded with the Jung-Karriere-Förderpreis 2024. I'm a clinician scientist working in the field of cardiac stem cell medicine that is based on human pluripotent stem cells that can be cultured in vitro and differentiated to any kind of somatic cell. We in the field of cardiology differentiate them to cardiac progenitor cells that give rise to smooth muscle cells, endothelial cells, and mostly cardiomyocytes. And we are particularly dependent on this technology because the adult human heart has, unlike many other organs, no resident stem cells that could be isolated and would help us to study the cardiac development and cardiac diseases. Those IP or human-induced pluripotent stem cells can either be obtained from the human blastocysts as embryonic stem cells or be obtained or generated since the advent of the iPS technology from any kind of somatic cells by reprogramming them back into a pluripotency stem cell state. And I highlighted already, we in our lab are differentiating them mainly to cardiomyocytes. Recently, the field has moved on from classical 2D culture models towards more complex models being 3D culture environments, cardiac organoid models, or multicellular constructs. And one of our 3D models that we are using in our lab is based on the idea to subject those in vitro grown cardiomyocytes to a surrounding that resembles native conditions as closely as possible. And for this reason, we came up with the idea to seeding those cells onto a native extracellular matrix scaffold that we obtain from native left ventricular heart sections when we wash out all native cells and end up with this extracellular matrix as a scaffold. And thereby we generate the so-called 3D engineered heart patches. With the help of our 3D bioprinter that was developed with our collaborator from the Hochschule München, we are able to homogeneously place the cells within this extracellular matrix, where they nicely have shown to integrate and show an increase and a maturation of their contractile performance, and also a matured phenotypes of their electrophysiologic behaviors like the effective refractory period and the force frequency relationship. And not only functionally, these cells perform quite well and mature, but also in terms of their function. They nicely align within the matrix and show thickened sarcomeric structures. So as these 3D culture conditions promote so-called the transition from immature cardiomyocytes towards a more mature phenotype, it gives us the chance to study developmental defects or to study the congenital heart diseases, because this is where we need to follow developmental processes from a transition to immature cardiomyocytes to cardiomyocytes. And one of the most common congenital heart diseases is the hypoplastic left heart syndrome, called HLHS. It is a severe cardiac disease that is characterized by a severely underdeveloped and unfunctional left ventricle and its adjacent structures, and patients usually die soon after birth. And we in our lab have generated patient-derived pluripotent stem cells from 3 different HLHS patients, and we subjected them towards this 3D culture where we have these heart patches. And we were really lucky and fascinated to see that these HLHS cardiomyocytes did not perform like the control cardiomyocytes, but they showed a very severe phenotype, which was characterized by a drop of contractile performance in this 3D culture system, and they have accumulated a multinucleation and an apoptosis. And with these findings, we were able to characterize 3 different or major hallmarks of the hypoplastic left heart syndrome. which are defects in cell specification, in their maturation, that ultimately lead to the apoptosis of left ventricular cardiomyocytes. So while we use these 3D engineered heart patches also for studying other diseases and other treatment approaches, we realized while we were cutting more and more left ventricles to generate this extracellular matrix that it would be extremely elegant to directly culture the native heart tissue slices and use it as a model for simulating and researching heart diseases and treatment options. Actually, our collaborator, Andreas Dendorfer from the Walter Brendel Institute in Munich, established a system that allows the cultivation of native heart tissue slices by subjecting them to a biomimetic culture conditions that is characterized by the application of a physiologic preload and a continuous electrical stimulation. And with this application, he has shown that it's possible to culture human heart tissue slices ex vivo for up to 42 days. And we in our group have adapted these culture conditions for large animal models, being pig heart slices and non-human primate heart slices. And this is how it looks in our incubator. I think you have seen it in the movie before, that they are in the incubator on a rocking board to ensure oxygenation, and we continuously have a readout of every single chamber that shows us the contractile force and the electrophysiologic behavior. As I said, these 3D heart tissue slices can be an ideal model to study therapeutic approaches, and this is what we did as part of a multidisciplinary study that was focused on the Duchenne muscle dystrophy. It's called DMD, and it is a very severe dystrophic disease affecting the skeletal muscles, where individuals usually are bound to wheelchairs in their teenage ages, and they also have a heart affection that is characterized by an end-stage heart failure and the development of a sudden cardiac death. In collaboration with our large animal group led by Christian Kupert, we were working with a transgenic pig that carried a humanized DMD mutation that is a deletion of exon 52, which then leads to a frame shift. And these pigs had a very severe phenotype, which was characterized by a very limited survival time. Using an AAV vector to reframe the mutation with a CRISPR-Cas9 technology, it was indeed possible to reframe the mutation, and we have seen that these pigs, after this treatment, have shown a prolongation of their survival time. But to understand the therapeutic effects of this reframing and gene editing, we generated heart tissue slices of these pigs to understand what is happening on a single cell level. And we were quite happy to identify that actually underlying calcium cycling defects caused arrhythmias in untreated pigs, but these were almost entirely abolished after the correction of the mutation. Ultimately, we thought that we have an expertise on iPS-derived cardiomyocytes, or the iPS technology, and we wanted to combine this with this ex vivo heart slice culture and to generate a chimeric heart tissue slice, which would consist of our iPS-derived cardiac progenitor cells seated on top of native non-human primate heart slices. Because we knew from our previous studies that native non-human primate heart slices would die up in the ex vivo culture after about 21 days. And we were really curious to see what happens if we place those cells, our cardiac progenitor cells, now on top of it. And it was quite fascinating and surprising to see that after 21 days, we did see an initial drop of contractile force, but we then saw a regain and a restoration of contractility that was significant already after 21 days and stayed up until 50 days when we decided to stop. And these results hinted towards the finding that the cells would really repopulate the native myocardium, which they not only functionally did, but we have also seen an electrical integration of our cells within the native myocardium, which we analyzed by point stimulation. And also in terms of structure, we have seen that our cardiac progenitor cells repopulated the native heart slice tissue and formed nicely elongated ventricular cardiomyocytes. So having a tool in hand that allowed us the analysis of cell-cell interaction and to study cells that obviously are capable to repopulate native myocardial, we were much more interested to investigate this in a disease-relevant context. So coming from a clinical perspective, the most common clinical scenario where we would need cell therapy to replace damaged heart and repopulate native myocardium is when a patient presents with a myocardial infarction. Thereby, we typically, or the patients typically present with a thrombotic occlusion of a coronary artery, which we can treat with a stent implantation, and we restore the blood flow again. However, due to this initial lack of blood and oxygen supply for the heart muscle, there is always some sort of cell death occurring. And some unlucky patients end up with a cardiac scarring and a heart failure. And we in the clinics treat these patients with a variety of medications, and there are also some devices available, but there is no causative treatment for these patients to really restore their capability of contractility. And for this reason, there is a lot of research going on to find new therapeutic strategies for heart regeneration. And the approaches are different from cell-free therapies to cell-based therapies. And I think I already highlighted that we are working in the field of cardiac cells obtained in vitro. And the idea is to use those cells to repopulate the injured heart. I have shown you some of our first experiments where we did not use fully differentiated cardiomyocytes, but rather their ultimate precursor cells, which are called cardiac progenitor cells. They're a much more plastic cell source that is still able to sense environmental cues and is still able to migrate. And we wanted now to investigate in our ex vivo culture if these cells are able to culture or to repopulate the injured heart. As a model, we developed an injury model that would simulate the myocardial infarction ex vivo. And we did this by placing a very defined cardiac injury using an RFA catheter. It is called radiofrequency ablation, and some of the clinicians here might know it because we usually treat arrhythmia patients with that method. But we took our ex vivo cultured heart slices to the EP lab and placed a very defined injury with this RFA catheter within our ex vivo cultured slice. And indeed, this turned out to be a very valid and standardized model to mimic cardiac injury and cardiac scarring, because cardiac fibroblasts were activated, and we did really see the formation of a collagen-rich scar in our ex vivo model, not only in terms of their structure, but also the function dropped after RFA injury and stayed low as we see it after scarring. I want to highlight the cells that were interested to investigate in this context. As I said before, we wanted to have not fully differentiated cardiomyocytes, but we're rather interested in cardiac progenitor cells that are differentiated towards cardiomyocytes, but we obtained them already at developmental day 6. And the cell line and differentiation protocol we used in this project was actually developed together with our collaborator Kylie Fu and Ken Chen from the Karolinska Institute in Stockholm. And the beauty of this cell line was that it carries an NKX 2.5 EGFP promoter, which basically just means that the cells would turn green as soon as it commits to being cardiomyocyte. And the experimental design was that we wanted to place the cells on one side of the tissue and compare our progenitor cells with cardiomyocytes. On the opposite side of the tissue, we generated this RFA catheter-induced cardiac injury. And then we just wanted to follow the cells and how they behave. Just briefly, we again achieved the cell placement, the selective placement, by using our bioprinting approach. Live imaging of these patches was really fascinating because we have seen that our cardiac progenitor cells indeed were able to start a migration directly towards this RFA injury already 1 day after the injury. And even at 4 days later, they enriched in this injury and repopulated this. And this phenomenon was exclusively present when we used our cardiac progenitor cells, but not when we compared them to cell seeding with differentiated cardiomyocytes. Also, the cardiac function with cardiac progenitor cells was quite surprising because we have seen that our HVPs really restored the contractile performance after injury, and this was only present when we used the progenitor cells, but not the cardiomyocytes. They also reduced the scar size. As you can appreciate from this immunofluorescent pictures, they nicely aligned in this RFA injury and reduced scar volume. And most importantly, they were also able to integrate in this scar because we performed point stimulation and have seen a conduction throughout the whole tissue to this RFA injury exclusively in the presence of the progenitor cells. So we were quite interested and also happy about our finding and wanted to understand what is really going on, why the cardiomyocytes land there and the progenitors, what is going on. So for this reason, we performed sequential immunostainings at different time points and were surprised to learn that not our cardiac progenitor cells were the first ones to reach the scar, but cardiac fibroblasts were the first ones to go there, and they were heavily repopulating the scar, but then at later time points they were kind of repelled from the upcoming cardiac progenitor cells, which were then the predominant cell type within the scar. So from all these findings and observations, we hypothesized that there must be some kind of chemical interaction of cardiac fibroblasts and cardiac progenitor cells. And we hypothesized that the HVPs, the progenitor cells, might be chemo-attracted by cardiac fibroblasts, and once these progenitor cells arrive in the scar, they repel the cardiac fibroblasts and end up with a fibroblast-free zone in the infarction. But to test this hypothesis, we needed to perform single-cell transcriptomic analysis of these slices, which we did by analyzing these patches at different time points after the injury. So at day 1, day 2, and later time points. And it was quite interesting already to see that the progenitor cells formed different clusters at different time points. And we have seen that indeed these progenitor cells are able to undergo different functional states in this process of cardiac repair, because at the beginning they are not activated, they are just sitting there and express cardiac progenitor cell genes. But then 1 day after injury, they go over to a sensing state, which we called, because they express cell activation and ECM genes. And then later on, after 48 hours, these cells really highly expressed cell motility and cell projection genes. So we call this a functional state of migration and counteraction, because ultimately they differentiated to cardiomyocytes once they landed in this injury region. For the sake of time, I will not talk about all the loss and gain of function experiments which we did to identify the signaling pathways going on from the cardiac progenitor cells and the cardiac fibroblasts. But I want to summarize the findings which we have identified, that the progenitor cells are indeed chemo attracted via CXCL4, CXCL12 interaction between fibroblasts and progenitor cells. And once these counteracting cardiac progenitor cells reach the injury, they highly express SLIT2, which interacts with ROBO1 expressed on cardiac fibroblasts, which leads to a repulsion of cardiac fibroblasts. And these findings allowed the conclusion that the cardiac progenitor cells are able to migrate specifically to cardiac injury and have antifibrotic activity during injury repair. So we were quite happy with these results, but of course we needed to prove whether, or to test whether this was only a phenomenon occurring in an ex vivo setting, which is why we chose to validate these findings in a large animal in vivo model. We chose, together with our collaborator from AstraZeneca, a large animal model in pigs, where the pigs were subjected to a myocardial infarction, and after waiting for 21 days, we have seen that they developed a big scar, and into the scar we injected our progenitor cells and waited for another 12 weeks to perform histology and analyze heart function by cardiac MRI. In this short video, you can appreciate that you see here the chunk of myocardial infarction of the pig, and in green our labeled cells that nicely dispersed and migrated within the native myocardium, and in red you can appreciate that we counterstained the cells for human nuclei to really be sure that these cells we're looking at are our cardiac progenitor cells. And indeed you can see that they accumulated within the scar. And looking more into the histology of these grafts, we have seen that the cardiac progenitor cells really formed nicely HVP-derived cardiomyocyte within the scar area. They elongated and they were even vascularized grafts. The cardiac function was also quite convincing, because we have seen that our progenitor cells do reduce significantly the infarct volume after 12 weeks, and they preserved the heart function after myocardial infarction, which was not present when we did not apply any treatment. These pigs showed a drop of their contractile performance of the left ventricular ejection fraction. Now, one of the biggest limitations of injected cells, and this is especially applying for differentiated cardiomyocytes, is the development of arrhythmias. For this reason, we have equipped the pigs that are in this experiment that I've just shown you with a biomonitor that would monitor their heart rhythm for the entire period of this analysis. And we have compared our analysis of the pigs with a published cohort of Romagnolo et al., which performed a similar experimental design as we did, but they used fully differentiated cardiomyocytes. And we have seen that already the number of pigs that developed a ventricular tachycardia, here called VT, was drastically lower in our HVP-treated animals than in the cardiomyocyte-treated animals. But more strikingly, the time that these pigs spent in VT, in arrhythmia, was significantly lower in our HVP-treated animals than in the cardiomyocyte-treated animals. So from what we know and what I've shown you so far, HVPs seem to be a great candidate for heart regeneration. And if you think of streaming them towards cell therapy, there are some endpoints that we already have seen, because they reduce infarct size and preserve heart function, and they form vascularized grafts. However, with the help of the Jung-Stiftung, I want to address further endpoints that are really key endpoints for clinical translation, which is the application route, because usually if in our studies that I've shown you, we have performed lateral thoracotomy, which would mean for patients a big mortality and morbidity risk. So we would need to find minimal invasive application routes. Further on, I have shown you some preliminary analysis on the arrhythmogenesis, but it's really essential to perform a head-to-head comparison of their arrhythmogenic behavior in vivo. And finally, I want to look at the graft performance in vivo. I want to track the cells, how they behave once they are injected in the body, and also look at off-target effects. And I think with these further milestones that we are addressing now in this project, we will be a further way down the road to the clinic, and then can think of many other questions that will arise in that journey, and then ultimately think of suitable clinical diseases and plan clinical trials. But until we are there, I want to thank everyone for listening, and of course my supervisors, Karl Ludwig Laugwitz and Alessandra Moretti, and the whole team. Of the people in the lab and the collaborators, and once again the Jung-Stiftung for this big help and trust that they're setting in me. Thank you very much.